Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the sixth lecture and we will learn about quantifying protein dynamics using FREP, that is fluorescence recovery after photobleaching, then FLIP, that is fluorescence loss in photobleaching, and FRET, that is fluorescence resonance energy transfer. So today mostly we'll focus on these three techniques, FREP, FLIP and FRET, which are generally used for quantifying the protein dynamics and the protein-protein interactions. So let us see why it is required to study protein dynamics. So here I'll show you one image of a GFP labeled protein. Okay, so this is a somatosensory cortex mouse brain slice expressing GFP. Okay, and here you can see that at the bottom you can see the neuronal cell bodies, and here these are the dendrites from the neuronal cell bodies, and some protein is GFP type. Okay, now by looking at this image, can you get an idea whether this protein is getting exchanged between these dendrites and the neuronal cell body, or they are not getting exchanged? The answer is no, right? By looking at one static movie or static image, we cannot say that whether the GFP labeled protein is getting exchanged between the neuronal cell bodies and the dendrites. So to get the idea about the dynamicity of the proteins, we need certain methods by which we can actually quantify the protein dynamics. So today we'll see one such method, that is known as fluorescence recovery after photobleaching, or FREP analysis. So this FREP analysis was established by Axelrod in 1976 to study the protein mobility or dynamics in living cells. So see, what are the methods that we'll discuss today for studying protein dynamics are mostly for the live cells, and the FRET that we'll discuss at the end. So FRET is used in even fixed cells as well. Okay, but let us focus on this FREP analysis first. And this method is used to measure the diffusion dynamics of proteins in live cells. So let me explain how this method works. So let's say this is a cell and you have labeled your protein of interest with GFP. So that protein is present in the entire cell as well as the nucleus. So you can see that entire cell is full of GFP labeled protein. But you don't know whether this protein is dynamic or it is static at that particular position. So let's say the protein molecules which are present at this particular position, they remain there forever, or they're actually exchanging with the nearby molecules. Okay, so how you can learn this thing? So for that, under the microscope, when you see the cells expressing GFP, using a high laser power, microscope allows you to bleach a small area in the cell. So let's say you will make one region of interest here, and you will ask the microscope to use 100% laser power or the full laser power to bleach these molecules in this area. Okay, so because of the excessive power laser light, the fluorescent proteins which are present in this particular area, they will be bleached. So you will see a cell like this, which the entire cell is shows green fluorescence except this area. But then you will image this cell at several time intervals, like 50 second, 100 second, 150 seconds. And what you will see, gradually this spot is getting recovered by the GFP molecules which are present in the surroundings. Okay. And within 150 seconds, you can see that now you cannot see that bleach spot in this cell after 150 seconds. So what does it represent? It represents that the proteins which are present in the surrounding are getting exchanged with this area, and that's why you cannot see the bleach spots after 150 seconds, because within 150 seconds those proteins have exchanged. So this indicates that this green your protein of interest, which is labeled with the GFP, is dynamic in nature. Now let's say below you can see an example of the slow diffusing protein. So here I have shown a comparison between a fast diffusing protein and slow diffusing protein. So let's say there are two proteins. You have labeled protein A with GFP, which is a fast diffusing protein, and protein B with GFP, which is slow diffusing protein. So how you can quantify the differences in their dynamic behavior? So we'll do the FRAP analysis for both the proteins in the same way, and you will measure the recovery. So you can see that the slow diffusing protein takes longer time to recover, like it takes 300 seconds to recover completely, whereas the fast diffusing protein gets recovered in 150 seconds. Okay, so this is how you can compare the diffusive parameters, diffusive property of proteins in live cells. Now here you can see intensity within the bleach spot, the curve for the intensity within the bleach spot. So suppose you are measuring the fluorescence intensity within this bleach spot. So how this curve will look like? So see, initially your intensity was high because there was no bleaching here, but then you bleached with a high laser power. So uh, uh, now the fluorescence intensity goes down to almost zero. Oh, and then you are seeing the recovery. So you can see that the protein which is recovering faster, the recovery curve will look like this. And for the protein which is recovering slower, you will see the recovery curve like this. So by looking at the pattern of this curve and the recovery, you can say that this protein is highly diffusive in nature, whereas this protein, which is shown by the orange line, is relatively slow in diffusion compared to the protein which is shown by the maroon color. So using this FRAP experiment, you can get to know about the diffusive property of the proteins, how dynamic they are in the cell, in the live cells, you can measure that. Okay. So here I have explained a typical FRAP curve. So typical FRAP curve looks like this. So this is the fluorescence intensity before photo-bleaching. Then because of the photo-bleaching, the fluorescence intensity goes down. And then you will take the image at several time interval. Okay. And you will see how fast the protein is getting recovered. So this is known as the recovery phase. This point is known as the photo bleaching point, and this is known as the pre-bleach condition, before bleaching or pre-bleach condition. Okay. Now, what kind of biophysical parameters that you can quantify using this FRAP curve? So see, here you can see that this recovery, you will never see the recovery back to the 100% of the original fluorescence intensity. So see, the original fluorescence intensity was at this level. Okay, and when you are seeing the recovery after photo bleaching, it will never reach to this stage, because there is some immobile fraction of molecules present at that location. So what happens, let's say in this figure, you see we bleached here. Now in this particular area, there were 20% molecules, let's say, they were bound to some structure in the cell. Okay. Whereas the remaining 80% molecules were just diffusing, were not bound to anything, and they were just diffusing. So those 80% molecules will recover faster. But this 20% molecules which are already there and they are bound to certain things, they lost the fluorescence, but that doesn't mean that the protein has gone from that area. Okay, protein is still bound to the same place. Only thing is, because of the high laser power, it lost its fluorescence. Okay, and that's why you can see the complete photo bleaching. But when you see the recovery, those 80% molecules which are just diffusing, they got exchanged, but those 20% molecules which are now dark, which are not fluorescent, they are still there. So that's why this recovery curve can never reach to the original fluorescence intensity, and this fraction is known as the immobile fraction, and this fraction is known as the mobile fraction. So using this FRAP experiment, you can get to know that what fraction of your protein is mobile fraction and what fraction of your protein is the immobile fraction. Also, you can also calculate the tau half. So tau half is the half time of fluorescence recovery. Suppose you have to compare the diffusion dynamics of two proteins, then you can see these curves, and then you can take you can extrapolate the line from the half of the fluorescence intensity to find out the find out the tau half value for both the proteins, and you can compare that protein A has a tau half of 30 seconds, whereas protein B has a tau half of 40 seconds. And also from this recovery curve, you can calculate the diffusion coefficient. So coming to some general considerations for FRAP experiments. So let's say you are setting up a FRAP experiment. So what kind of practical considerations you will keep in mind? So the first thing is optimize the sampling time after the bleaching event, as recovery for fast diffusing protein takes a few seconds, whereas the recovery of the slow diffusing protein takes a few minutes or hours. Okay, so what it is saying, let's say you are setting up the FRAP experiment. So how you will decide that you will image after bleaching, you will image at every 50 millisecond, every 50 seconds. Okay, let's say this is a fast diffusing protein, so it may recover in 300 seconds. But let's say there is some slow recovering protein. Okay, so it may take, let's say, 10 minutes to recover. So in that case, you have to image up to 10 minutes. But in that case, you will not image at every 50 seconds, because each imaging also creates some kind of a some level of a photo bleaching. Okay, so you should not image unnecessarily. But let's say your protein is slowly diffusing protein, then you will keep this time interval, let's say every 5 minutes, 5 minutes, 10 minutes, 15 minutes, 20 minutes, 25 minutes, and 30 minutes. So you can see whether in 30 minutes your protein is getting recovered or not. Whereas for fast diffusing protein, you have to set the time interval so small, like 50 at every 50 seconds if you image, and then you can see the recovery within 300 seconds. So when you are working with a new protein, you have to optimize this thing. First, you will try with the very short time interval, like 50 seconds, and see how fast it is recovering. If it is not recovering, then keep little longer time interval, like image at every 5 minutes, image up to 30 minutes, and see whether the protein is getting recovered or not. So that thing you have to optimize, that what time interval you will image and for how long you will image. So this experiment will finish in 6 minutes, because of the 300 second you are getting complete recovery. But let's say if you are working with a slow recovering protein, then you may have to see this recovery up to, let's say, 1 hour. So that experiment will taking one FRAP image will take 1 hour. Okay. The second consideration is optimize imaging parameters to minimize photo bleaching outside of ROI during the recovery phase. So while setting up this experiment, you have to set the microscope parameters, the exposure time, the power of the illuminating light source. Okay, you just set it to the lowest power and lowest exposure time, so that it should not create unnecessary photo bleaching. So let's say here the photo bleaching took place with 100% laser power, because for photo bleaching we use a high laser power. But here, for taking this images during the recovery time, our intention is not to uh, create more bleaching. Okay, so for that purpose, you have to optimize your laser power as well as the exposure time in such a way that you can just see the GFP fluorescence, but it should not be overexposed, or higher light laser power should not be used, so that it creates unnecessary photo bleaching. Then the third thing is avoid pixel saturation. So this is a quantitative experiment. So you do not want that some of the pixels were saturated with GFP, so they cannot give you the proper quantitative intensity information. So you have to make sure that your camera pixel, or whatever the if you are using the confocal, then your pixel should not be oversaturated with the photons, and you should reduce the laser power or the exposure time accordingly, so that the pixel saturation should not take place. The fourth parameter is optimize the size of your photo bleach area, that is region of interest, because soluble proteins need a larger array. So let's say in this cell we use such a small area. Okay, so what so even how much area you will bleach, that also you need to optimize. So it is mentioned that the soluble proteins need a larger ROI. So let's say if you know that your protein is diffusing fast, then you should make a little larger ROI, so that you will get enough time to measure the recovery. But let's say if you will make a very tiny spots, and you will bleach that spot, and that spot may get recovered within 50 seconds. So even you will not be able to measure the recovery. Okay, so if you bleach a little bigger area for the fast diffusing protein, then only you will have some time to measure the recovery. The next thing is empirically determine photo bleaching conditions. Use a minimum number of laser iterations. So what it tells, generally microscope software allows you to go for multiple iterations to bleach this area, because sometimes it is possible that even you are using 100% laser power with uh, uh, let's say 30 millisecond of pulse. With one pulse you may not able to bleach this spot completely. You may need several iterations, like you may need 5 iterations of 30 milliseconds to bleach this spot. Okay, so try to minimize the iterations. And for minimizing the iterations, what you can do is you can just use your laser at 100% power to avoid to reduce the number of iterations to bleach this area. It should not be like you use your laser with 30% power, but uh, go for prolonged bleaching. You bleach your sample for 10 iterations, for even by giving 100 or 200 millisecond pulse. Okay, so keep your laser pulse for shorter duration as much as possible, generally 30 to 50 milliseconds. Use laser at 100% power and try to bleach the area as quickly as possible. The sixth practical consideration is continue imaging until the recovery plateau is achieved. Continue imaging until the recovery plateau is achieved. So let's say in this experiment, what will happen if I just image up to 100 seconds? If I image up to 100 seconds, I am not imaging until the full recovery. So it will not give me the full details about the recovery. Okay. So you have to image until you can see the plateau in this recovery. So initially we will see that fluorescence intensity is coming back. It is increasing. But so you have to image until this kind of a plateau is reached in fluorescence intensity. GFP is still the most commonly used fluorescent protein for FRAP. Okay, so generally for FRAP analysis, GFP is most widely used protein because it is easily bleachable. And generally fluorescent proteins that are photostable are not good for FRAP. So in the last to last class, we learned about several fluorescent proteins which are more photostable. For an example, TAG RFP or neon green. Those are the photostable versions of the GFP. So for FRAP analysis, you should not use such kind of fluorescent proteins which are photostable, because we want to, this is a bleaching based application. So we want the protein to be bleached. And that's why GFP is most widely used fluorescent protein for the FRAP analysis. And here you can see, I have mentioned some of the relative diffusion rates for GFP in water, GFP in cytoplasm, GFP MHC class 1 in endoplasmic reticulum, and GALT GFP in Golgi. So just to give you some idea about the diffusion coefficient, the unit of the diffusion coefficient is micrometer square per second. So you can see if the free molecules are there, you will get the diffusion coefficient of around 90. But if they are constrained in the cytoplasm or the nucleus, then you can see the diffusion coefficient of around 25. And for different taggings or based on the protein function and localization, the diffusion coefficient may change. So this diffusion coefficient you can measure from the FRAP curve by taking the slope of this recovery. Now on this slide, I will show you how to normalize the FRAP curve. So FRAP is highly quantitative method. Okay, so for better quantification, you need to normalize the FRAP curve. And why that normalization is essential, that I will tell you. So the first thing is normalization for photo bleaching occurred due to imaging. So why normalization is required? So the first point is because you are imaging after photo bleaching for several frames, right? So that imaging will also photo bleach your fluorescent molecules. So that's why you need need to normalize your fluorescence intensity of the bleached spot with that of the unbleached area. Okay, and gap ratio is the loss of fluorescence due to FRAP bleaching. Okay, because you are using 100% laser power and you are intentionally bleaching a small fraction of molecules, so that will reduce the overall brightness of your sample. So let's say here I have shown one typical FRAP curve and I have shown the other region of interest that are used for the normalization. So for any FRAP experiments, you should measure, quantify the fluorescence intensity of these 4 regions. The first one is known as the FRAP ROI. So here you can see that in this sample, I have shown the FRAP ROI in red color. So let's say in this sample, this is the area where you will do the photo bleaching. So here you can see in this image, it is photo bleached, and then you will measure the recovery in this area. So that is your FRAP ROI. And if you see the fluorescence intensity of this FRAP ROI, it looks like this, that initially the intensity was at this level, then you photo bleached, so intensity went down, and then you are seeing the recovery in the fluorescence intensity. So this is a typical FRAP curve. But for the normalization, you will make one reference ROI at the unbleached area. So see, in this sample, they are not bleaching this bright white uh, area. Okay, so this there we can use as a reference ROI. So how the fluorescence intensity of this reference ROI changes over time, because see, we are taking so many images, so every time we are introducing some additional photo bleaching. Okay, so this blue ROI and the fluorescence intensity of blue ROI will show the gradual decrease in the fluorescence intensity due to imaging. The third ROI is the base ROI. Base ROI means the background fluorescence intensity. So here the background is dark, okay, but in some cases you may find that there is some fluorescence in the background. So for that you will measure the fluorescence intensity of this background, and that is considered as the base ROI. And here you can see the base ROI is, the fluorescence intensity of base ROI is almost negligible. And the fourth one is the non-FRAP cell ROI. So this is another cell in which we are not doing any kind of photo bleaching. So we will measure the fluorescence intensity of this cell to see how it reduces over time, just to consider the photo bleaching due to imaging. Okay. So we have to measure the fluorescence intensity of these four regions, and we have to normalize it with the actual FRAP ROI intensity. For details on calculation and kinetic modeling for the FRAP curve, you may visit this website. There is a nice description on actual quantification of the FRAP data. This is a slightly different version of the FRAP, which is known as inverse FRAP. So generally in the FRAP experiment, you learn that this is a specific area which is photo bleached, and then you measure the recovery in this area. Okay, and your FRAP curve looks like this. In inverse FRAP, what you do, you bleach everything except this small part. Okay, and then you see that how this small part fades up. Okay, so this recovery curve will look like this. Initially the intensity of this area was high, and then gradually it decreases like this. So this is known as the inverse FRAP. So for some kind of application, let's say here the inverse FRAP has been done in this kind of structure where some area is very bright and the other area is has the weak fluorescence. You can do inverse FRAP, like you just bleach everything except this bright spots in the center, and then you see how the fluorescent proteins from this bright spot are diffusing into this area. So you can see in 60 minutes this bright spot become faint, and the fluorescent molecules which are present in this area, they migrated to the other areas. Okay, so in some such kind of applications, the inverse FRAP may be useful. So coming to fluorescence loss in photobleaching, which is known as FLIP. So this technique is used to check the continuity in cellular compartments and reveal the protein subpopulation bound to non-mobile macromolecular structures. Okay, so how this technique works? So let's say this is a cell, and what you will do, you will make this region of interest for iterative bleaching. So in the first image, in the second image, you will bleach here. So all the fluorescent molecules are bleached in this area. Then again, you will allow some time to recover the molecules in this area. And then again, you will bleach. Then again, you will allow some time to recover the molecules in this area. Again, you will bleach. Again, you will allow to recover the molecules. Again, you will bleach. So we'll do iterative cycles of bleaching, and you will give some times for recovering the protein. And eventually, after several such cycles, what you will see that the fluorescent molecules in entire the cell are bleached, but some molecules in the nucleus, they remain fluorescent. What it indicates that these molecules which are in the nucleus, they are not exchanging with the cytoplasm, so that when you bleach the molecules in the cytoplasm, this molecule still remain fluorescent. Okay, so this technique is used to see the continuity in cellular compartments. So whether, if you want to see that, whether the protein molecules which is there in the nucleus, it is getting exchanged with the cytoplasm or not, you can use this technique. You can do the iterative bleaching in the cytoplasm and see whether fluorescence is lost from the nucleus or not. Similarly, here is another example. So let's say uh, this is an example of the flip only, and you can see this is a continuous compartment. There is some cell organelle, okay, which is continuous, and here you can see there is a discontinuous compartment, and you want to see that whether this compartment is continuous or discontinuous. So what will happen? So this red color area is your bleaching spot. So you will do iterative bleaching at this red spot so that all the fluorescence molecule which are there in this structure, they will be bleached. Whereas here, if you do the iterative bleaching at this red spot, all the molecules in this compartment are bleached, but the fluorescent molecule which are there in this compartment is different. So they are not getting exchanged with this compartment, and they will still give the fluorescence. Okay, so this is how you can differentiate between continuous compartment and discontinuous compartment by doing iterative photo bleaching in one small area within the cell. So here are some practical considerations for the flip analysis. So the first thing is, what if the bleach box were twice as large? So in our experiment, we use the bleach box of this size. But what will happen if I make the bleach box little larger so that half of the cell is bleached? So in this case, you can see there will be this kind of differences in the fluorescence recovery. So the rate of loss of fluorescence depends critically on the size of the bleach area, the shape of the bleach area, and where it is placed within the bleach area. So if it is a fast diffusing protein, then it's better to go for little bigger bleach area. But if it is a slow diffusing protein, it is better to go for the small bleach area. Just like the FREP experiment. Another practical consideration, you should give enough time for sufficient re-equilibration. So let's say this is iterative bleaching, right? So this is the first bleach in which you bleached all the fluorescent molecules in this area. Now you are, let's say, giving 5 minutes for the recovery before bleaching it again. So in 5 minutes, let's say the protein molecules are properly re-equilibrated in this area, and then you can bleach again. Okay, but if you go a little faster, you bleached here, then immediately within, let's say, 50 seconds, you are bleaching again. Then you are not giving enough time for recovery. So what will happen? This area will not be re-equilibrated with the fluorescent molecules. So repeating bleaching before re-equilibrium is unnecessary, and it increases the risk of phototoxicity and photo bleaching of the signal. So faster bleaching is unnecessary, and it increases the risk of phototoxicity and photo bleaching of the signal. So you should give ample time for the re-equilibration, and all these things can be optimized by trial and error. So if you are working with a new protein, it's not that your experiment will work in the first shot. First you have to optimize these parameters, that how fast my protein is diffusing, so that I can set the time interval so that sufficient re-equilibration can take place. Also, cells have an innate capacity to destroy the radicals, and discontinuous bleaching may decrease long-term phototoxicity of the flip experiment. So generally, this is known as the continuous bleaching. Okay, so you are continuously bleaching and imaging, bleaching, imaging, bleaching, imaging, bleaching, imaging. Other ways, bleach, image, and then leave some time for equilibration. Bleach, image, leave some time for equilibration. Bleach, image, and give some time for equilibration in between. The best way to do this kind of flip experiment is go for discontinuous bleaching and imaging with a lag between iterations to minimize photo damage. So instead of a continuous bleaching, if you go for this kind of discontinuous bleaching, then it reduces the photo damage, because cells have an innate capacity to destroy the radical. So in this discontinuous bleaching, you are giving some time to the cell so that cell can destroy the free radicals produced during this photo bleaching. Okay, so this is the best way of doing flip experiment, in which you go for the discontinuous bleaching, then imaging, and there should be some lag between the iterations. So this is all about flip, and now let us move on to the next method, which is known as FRET analysis. FRET stands for fluorescence resonance energy transfer, or Foster resonance energy transfer. So this is a mechanism describing energy transfer between two light sensitive molecules, which are the chromophores. A donor chromophore is in excited state transfers energy to the acceptor chromophore through non-radioactive dipole-dipole coupling. Okay, so let me explain you this phenomenon using this diagram. So let's say this is protein A and B are my protein of interest, and I want to see whether this protein A interacts with protein B or not. Okay, so whether there is protein A and B, they are in close proximity or not. So what I will do, I will tag this protein A with the cyan fluorescent protein and protein B with the yellow fluorescent protein. I will typically use this FRET pair, this pair of fluorescent proteins. Why? Because you see their excitation and emission wavelength. So for CFP, the excitation wavelength is 433 nanometer and the emission wavelength is 475 nanometer. For YFP, the excitation wavelength is 475 nanometer and the emission wavelength is 528 nanometer. So you can see the emission wavelength of this fluorescent protein is same as the excitation wavelength of this fluorescent protein. So what will happen if these two proteins are placed close to each other? Then the emitted light from this protein can excite this fluorescent protein, and it will also give you the fluorescence in 520 at 528 nanometer, which is yellow in color. Okay, so if these two proteins, protein A and B, they are interacting or they are in close proximity, then FRET will happen. This phenomenon is known as FRET, in which the energy is transferred from one fluorescent protein to the another fluorescent protein. Okay, if the emission wavelength of this protein is same as the excitation wavelength of this protein, then if these two proteins are close together, then they can transfer the energy from one to another protein. And because of that, what will happen? You will use the excitation light for CFP, but you will see the emission in the YFP channel instead of CFP channel. CFP emission is blocked because that energy is getting transferred to YFP, and you will see the yellow color fluorescence. So it will look like this, like you are expecting a cyan fluorescence here, but you will not see cyan fluorescence, but you will see the yellow colored fluorescence. So this phenomenon is not known as FRET, fluorescence resonance energy transfer, because it is basically the energy transfer between these two fluorescent proteins. So this FRET is used to check protein-protein interactions, protein-DNA interactions, and protein conformational dynamics. So why these two pairs of fluorescent proteins are used, CFP and YFP? Because if you see, the emission wavelength of CFP has quite a bit of overlap with the excitation or the absorption wavelength of YFP. So this is known as spectral overlap. So because of this spectral overlap, this CFP and YFP is the best FRET pair, which is widely used for the FRET analysis. Nowadays, there are several other FRET pairs have been developed. So you can see here the donor and acceptor FRET pairs have been shown. Now this FRET is a phenomenon. This energy transfer can only take place if these two molecules are very close, means within 10 nanometer distance, then only the energy can be transferred from one fluorescent protein to other. If the distance is more than 10 nanometer, the energy transfer is drastically reduced. So you can see that here is the list of different fluorescent protein which are used as a donor and acceptor in the FRET analysis, and this is the Foster distance in nanometers. Okay, so if you use these two fluorescent protein, like let's say CFP and GFP, okay, then if the distance between protein A and B is less than 4.7 to 4.9 nanometer, then only you can see the FRET analysis. Then you can get the FRET signal. Okay, if you are using this Psi5 and Psi3 dye as a FRET pair, then if your distance is greater than 5 nanometer, then also you can see the FRET signal. Okay, so in FRET analysis, this protein is known as donor because it is emitting the light which is being absorbed by this protein. So this is known as donor and this is known as acceptor. So you will see that here the list of donor and acceptor. So these are basically two fluorescent proteins which are used as a donor and acceptor in the FRET analysis. And here their Foster distance is mentioned. So if your two molecules are within this proximity, then only they will give the FRET signal. So most widely used pairs are CFP-YFP and GFP-MRFP. Very promising pairs M Turkish YFP, Clover MRB. And these are some other pairs are also very promising like EBFP, GFP and several other pairs which are shown here. And here some recommendations for the fixed cells. So if you are doing FRET analysis in the fixed cells, you should go for Psi3 Psi5 or Etto 550, Etto 647, Alexa 555, Alexa 647. These are the best donor and acceptor pairs for the fixed cells, whereas these are the fluorescent proteins, so they are generally used for live cell imaging. So see, this FRET technique can be used in live cell as well as in fixed samples. Okay, so if you are going for the live samples, you should use some fluorescent proteins. If you are going for the fixed samples, you can use some of these dyes for the FRET analysis. So this is a little advanced technology based on the FRET, which is known as single molecule FRET, and it is used for studying protein conformational dynamics. Okay, so here you can see, let's say these are the two lobes of the protein, and this is basically you will see uh, uh, this is periplasmic binding protein of E. coli and it has two lobes. So when it binds to the ligand, it it is converted into this kind of closed conformation. So both the lobes will come in close proximity with each other. So if you want to see that the dynamics between this open and closed conformation, what you can do is you can label one lobe of the protein with Psi5 dye and other lobe of the protein with Psi3 dye, and then you can measure the FRET, that how frequently these two domains are coming in close proximity with each other. So you can measure that FRET. So when they are in close proximity, the FRET will happen and you will see the signal in the FRET channel. So you will see a peak. When they are far away, you will see a drop in the fluorescence intensity. So this is how, using this single molecule FRET, you can quantify the protein conformational dynamics. So coming to some of the practical considerations for the FRET analysis. So FRET can be measured in live cells as well as in fixed cells. Incorrect orientation of chromophores may result in absence of FRET despite their close vicinity. So it is possible that, see here, let's say currently protein A and B, they are interacting in the right orientation and that's why you are seeing the FRET. Okay, but suppose if they interact in this orientation, okay, then it is possible that this CFP and YFP may not come close together, and in that case you may not see FRET signal even though protein A and B are interacting, but they are interacting in a different orientation. Okay, so this CFP and YFP will not come close to each other. So sometimes because of this kind of orientation problem also, it may give you false information that it will not show you the FRET signal despite being the fact that the proteins are actually interacting. So this FRET data needs some kind of a confirmation by some other methods. Then some of the controls which are being used for the FRET analysis. So positive control. So this kind of plasmids are commercially available in which the protein of interest or any other protein is dual labeled like CFP and YFP. So here CFP and YFP are in the close proximity because they are the fusion proteins. Okay. Then the negative control, you should make the strains for negative control in which only the protein of interest 1 is labeled with CFP or protein of interest 2 is labeled with YFP in different strains. Okay, so that you can use as a negative control. And the experimental strain will have protein of interest 1 labeled with CFP, protein of interest 2 labeled with YFP. Basically A with CFP, B with YFP in the same strain. Okay. Now, how to measure FRET? So there are three ways of measuring this FRET. Okay, the first and the most simplest method is known as acceptor photo bleaching. So how it is measured? So bleaching of the acceptor increases the fluorescence intensity of the donor. Okay, so what it shows, coming back to this diagram. So let's say here what we discussed, that uh, consider that this protein A and B are interacting, and that's why the CFP and YFP are in close proximity. So if you excite this CFP at 433 nanometer, you will see fluorescence in 528 nanometer channel. Okay, but you will not see the CFP fluorescence like 475 nanometer light, you will not see. Okay, now to measure whether these two proteins are interacting or not, what you can do is, the first method is known as acceptor photo bleaching. So this YFP is the acceptor. So you photo bleach it. Okay. So if you photo bleach it, then it cannot accept the light, emitted light from CFP. Okay. So what will happen? It will increase the fluorescence in the CFP channel. Okay. So ideally, when you are exciting the CFP, you are seeing the fluorescence in the YFP channel. But now you have bleached YFP. So what will happen? It cannot accept the light from CFP, and it will increase the fluorescence in the CFP channel at 475. The second method of measuring FRET is sensitized emission. So here, measure the emission intensity of the acceptor by illuminating donor. If the distance between donor and acceptor is less than 10 nanometer, you will see acceptor fluorescence. So this is again a most simplest way, like you excite donor with 433 nanometer and quantify the emission at 528 nanometer. Whether you are seeing the fluorescence in yellow channel, if you are seeing the fluorescence in yellow channel, that means they are close together. Or if you are not seeing the emission at 528 nanometer, that means they are far away. Okay, so that is known as sensitized emission. And the third way of measuring the FRET is lifetime measurement. FRET efficiency can be determined from the change in the fluorescence lifetime of the donor. The lifetime of the donor will increase in the presence of the acceptor. So you will measure the fluorescence lifetime of the donor. So here CFP is the donor. So if this YFP is in close proximity, the fluorescence lifetime of this donor increases. Whereas if it is not nearby, then the lifetime of this donor decreases. So this is another way of measuring the FRET. So there are three ways by which you can measure the FRET. So sometimes it depends on what kind of features are available on your microscope, whether you can go for sensitized emission or acceptor photo bleaching or lifetime measurement, because all three need some different kinds of hardware components or even the software components in your microscope. So depending on the availability of those hardware components and the software components, you can measure FRET. With this, let us end today's class, and in the next class, we'll discuss about illumination strategies like epifluorescence illumination, turf and halo illumination, confocal illumination, light sheet illumination, and two photon illumination. So we'll discuss about different illumination strategies in the next class. Thank you.